everyone. I'll just give a brief introduction. So I'm Nicola Oakenfull from Myeloma New Zealand. Just like to welcome you to the session today with the Mulligan Institute. I've got Dr. Rachel Perret and Puja Paudel. They're going to both present and they will both introduce themselves as well. This recording, if you end up not being able to watch all of it, we will put it up on our YouTube page, possibly after Christmas. But yeah, just like to really thank Rachel and Puja for today. If anyone has any questions, then you can pop questions in the chat or the Q&A along the bottom if you're on a computer, and if you're on your phone, hopefully you'll be able to find them. And yeah, just ask questions and we'll leave time for questions at the end. Right, I'll hand over to you both.
All right, thank you, Nicola, and thank you to Myeloma New Zealand for hosting this webinar. Tēnā koutou katoa, ko Rachel Perret aho, ko hui te rangi te whakapaparanga mai, engari ko o tōtahi te whenua tupu. So my name is Rachel Perret. My family come from Switzerland, but I was born and raised in Christchurch, and I am the team leader of the CAR T-cell research program at the Mulligan Institute of Medical Research. Today, along with my colleague, Dr. Puja Paudel, we're going to tell you a little bit about the research that we do with CAR T-cells at the Mulligan and specifically about myeloma as well. Firstly, an introduction of the Mulligan. We are New Zealand's world-class independent biomedical research institution. So we're a registered charity, and in effect, we're owned by New Zealand for the benefit of all. We're located in Wellington, up on the hill in the University of Wellington Kelburn campus, and we have about 130 staff and students at the moment. Our focus is on making breakthrough discoveries in immunology and immunotherapy, so basically understanding more about diseases that affect New Zealanders and designing better therapies for that. So our main focuses of research and discovery are allergy, asthma and inflammation, also infectious disease, and cancer, which is what we both work on. So in the field of cancer, we're exploring new ways to train the immune system to recognize and fight cancer and to create safer and more effective treatments. And so in 2013 already, Science, which is one of the biggest scientific magazines in the world, named cancer immunotherapy, so using the immune system to fight cancer, as the breakthrough research discovery of the year. And since then, the field has been growing with immune therapies such as CAR T-cells, revolutionizing cancer treatment, as they're a lot more precise and focused than things like surgery and chemotherapy. So how CAR T-cell therapy works is that patients give a white blood cell donation, which is very similar to giving a regular blood donation. And from their blood, we separate out the T cells, which are sort of the killer and helper cells of the immune system. Their usual role is to fight off things like infections by bacteria and viruses, but they're not very good at recognizing cancer because cancer comes from our own cells that have mutated, and it makes it hard for the immune system to recognize them. So to get around that problem, we reprogram the T cells in the laboratory using a viral vector that allows us to give the T cells a new gene for this CAR, which is a receptor, or sort of imagine a little claw on the surface of the T cells that allows them to latch on to cancer cells and attack and kill the cancer. So once we've made those CAR T cells in the lab, we grow them into really large numbers, up to 100 million cells for each patient. And then we give patients a very low dose of chemotherapy to make some space in the blood for the CAR T cells, just before giving the CAR T cells in just a single small injection. And so you might be wondering, what is a CAR? It's actually an acronym for the term chimeric antigen receptor. And the chimera was a Greek mythological creature which has the head of a lion, and that's kind of like this external antigen or tumor recognizing part of the CAR T cells, and that's on the outside of the cell and interacts with the tumor. And then, like the chimera had the body of a goat, our CAR has this sort of stalk or stem that connects the outside and inside parts of the cell through the membrane, that's shown by these little beige dots here. And then one of the most important parts of the CAR is this internal signaling protein or domain called CD3 zeta, which comes from the natural T cell receptor and is the sort of key activating signal that tells the T cells, after seeing the cancer with their receptor, that they need to get activated and to attack the cancer. And so CAR T cells have evolved over the years since the late 1980s when they were first developed, from this first generation, which I've shown you, which is quite simple and has only the basic building blocks, to a second generation, which has been modified to incorporate a second co-stimulatory or co-activation protein. And that just makes the CAR T cells a bit more powerful. And the CAR T cells are actually used in the clinic in other countries are all of the second generation CAR T cell type. There has been since then a third generation of CAR T cells, which basically involves adding a third signaling protein or a second co-stimulatory or co-activating domain. And those are the kind of T cells that we're using at the Mulligan in our current ENABLE clinical trial for lymphoma. It hasn't stopped there. There's also a fourth generation of CAR T cells where they don't have extra signaling proteins inside the cell, but they have additional gene inducers that allow us to give additional functions or boost the activity of the CAR T cells further. And these are a bit more imaginatively named, such as Armored CAR, Turbo CAR, or TANKs, which are actually a project that's being run here at the Mulligan by the Ian Herman's lab. So there are a lot of different CAR T cell trials going along around the world. This is actually a little bit out of date already because it was from a scientific paper last year, and the authors accidentally left New Zealand out of their map. So I have adapted the image for them and added New Zealand with our one ENABLE CAR T cell clinical trial that's going on at the moment. So moving on a little bit now, more to a focus on multiple myeloma and how immunotherapies are used for this disease. So firstly, for those who might not be familiar with myeloma, it's a cancer of mutated immune cells or plasma cells, and it starts in the bone marrow. This is one of the most common hematological or blood cancers worldwide, and actually New Zealand has the highest global incidence of this cancer. There are even higher rates also in Māori and Pacific peoples, both in terms of incidence or number of cases and also mortality rates. So this is an important health problem for New Zealand. And we hear from patients and clinicians, and we ourselves, the scientists, want to push for access to better treatments here in New Zealand. So overseas, there have been CAR T cell studies, both in clinical trials and in the real world setting, that are showing really promising results. But unfortunately, we don't have access to those therapies yet in New Zealand. And in addition, some of the myeloma CAR T cell trials or data are showing that the long-term responses are not as high in percentage as for some of the B cell cancers that are also treated by CAR T cells. So there's room for improvement. This is a little bit of a history of the treatment landscape in the United States. So this is data from the FDA of when different myeloma therapies were approved, and some of those therapies are available in New Zealand and some are not. What we're going to focus on today is the CAR T cell therapies, highlighted here in teal, ide-cel, also known as ABCMA, or cilta-cel, which is also known as CARVYKTI, and the BiTEs, or the bispecific antibody therapies, of which there have been 3 approved, and one of those has already been involved in a clinical trial in New Zealand, the elranatamab, I believe it was last year in 2023. So I've already explained to you how CAR T cells work. They have a bit of an antigen recognizing receptor from an antibody on the surface of the CAR T cell, and then some activating signals within the cell. And that whole cell therapy is transferred into patients as the treatment. Bispecific antibodies, or BiTEs, are a little bit different. They are made up of 2 different antibody antigen recognizing parts. One of those that attaches to the T cell through the CD3, which is the T cell activating signal. And then there's a linker or a connector that connects a second antibody that recognizes the myeloma cancer. And so this antibody could be the same in a CAR T cell or a BiTE, or it could be different. The main difference between the therapies is one is a cell therapy and one is more like a standard drug. And so as I've pointed out, there is that first difference where CAR T cells are a living drug. And so one single injection could be curative in the case of a successful therapy, whereas bispecific antibodies act more like a regular drug in that you have to have multiple continuous injections to keep the levels high in the blood. One of the downsides of making individual patient therapies out of their own patient cells is that this is a bespoke product that has to be made for each patient, and it can take several weeks to produce. Whereas bispecific antibodies are an off-the-shelf therapy that's ready to inject at any time, and so it allows faster treatment of patients. There's sort of very similar targets for the CAR T cell therapy and the bispecific antibodies. Both of those can target BCMA, and the approved bispecific antibody or BiTE therapies can also target another myeloma antigen protein, GPRC5D. Possibly what's most interesting to us is how these therapies impact patients. And so CAR T cells have shown higher efficacy or complete response and overall response rates in terms of the tumor mass decreasing, particularly for cilta-cel or CARVYKTI, whereas BiTEs have lower toxicity rates. And this is particularly relevant for cytokine release syndrome, which is a general inflammation in the body, or immune cell associated neurotoxicity syndrome, which is localized inflammation in the brain. And both these side effects can be quite severe in CAR T cells. So what we really want to do is bring the benefits of both those two therapies together in new and improved future therapies. So this is just an example of CARVYKTI. So this is the leading current immunotherapy for multiple myeloma. And you can see here in the green lines that the CARVYKTI has a better progression-free survival rate than the competing product ABCMA. But in both of those cases, after an initial response rate that can be quite high, responses can fall off over time. So we do need to want to try and improve those. So that's where our CAR T cell therapy research at the Malaghan Institute comes in. We want to work to make better and safer therapies in the future. And so one of the methods we've taken to do this is to develop with some colleagues in China at the Guangzhou Institute of Biomedicine and Health, a third generation CAR T cell targeting CD19. So that's currently used in our lymphoma clinical trials. That has this additional activating domain. So again, we have this antibody fragment that recognizes CD19. That's going to be the outside part of our CAR that recognizes the cancer. And inside the cell, we have this standard CD3 zeta T cell signaling protein, as well as the co-activating protein CD28. But we've also added this additional TLR2 protein, and this stands for toll-like receptor 2. And in the immune system, this protein has a natural role detecting microbes such as bacteria and viruses and activating the immune system. So it's quite a good protein to boost CAR T cell therapy further. So showing you a little bit of the data from our lab, we've compared our third generation CAR T cells in the orange with second generation CAR T cells, which are like the ones that are licensed for clinical therapy overseas, just to see how well they're able to control the growth of a tumor that expresses the CD19 antigen. And we do this by taking microscopy photos of the mixed together tumor cells and CAR T cells at each of the little time points indicated by the symbols on the graph. And as the experiment progresses, we put additional tumor cells into the co-culture so we can see if the CAR T cells still remain good at killing the tumor. And you can see that normal T cells don't control the tumor growth at all. The tumor sort of gets bigger and bigger. With the blue line, which is the classical or the leading CAR T cell therapy that's approved internationally, there's some initial control of tumor growth, and this experiment was only done in the laboratory. Obviously, things might be different in patients, but eventually the CAR T cells lose control of the tumor. Whereas in the orange line, our third generation CAR T cells with the TLR2 signaling domain are doing a better job at preventing the cancer from growing. So this was a really good result. But we also wanted to test some other aspects of CAR T cell behavior, and that is their ability to produce cytokines or chemical factors that cells use to communicate with each other. And these particular cytokines, interferon gamma and GM-CSF, are important because they're involved in T cell activation and activation of other immune cells. But if there's too much of these cytokines, it can lead to those severe side effects that I was mentioning before, the CRS or the ICANS, inflammation systemically in the body or in the brain. So we were quite surprised, but encouraged to see that the orange graphs here are smaller than the blue ones. And so there's significantly less of these inflammatory cytokines with our third generation CAR. So we moved then into a phase 1 clinical trial of these same third generation CAR T cells in patients with relapsed or refractory B-cell non-Hodgkin lymphoma. And this trial was led by our clinical director, Dr. Rob Weinkove, and was performed at Wellington Hospital. So what we did was we enrolled patients and then took their donated white blood cells, and we produced CAR T cells from them in the labs at the Malaghan Institute. And this is a process that took about 4 to 5 weeks. And towards the end of the clinical trial, we were actually able to automate the cell manufacturing process to make it more efficient. Patients then get a PET scan to grade their disease. And then after the low dose of chemotherapy, just a single injection of the CAR T cells, after which their disease or cancer status is monitored over time. And so just to give you a sort of an overview, we were able to get quite a diverse group of patients in terms of age, ethnicity, and different lymphoma types. And most of the patients had had many lines of prior therapy before they got to having CAR T cells. And despite the trial being centered in Wellington, we actually were able to recruit patients from a lot of areas throughout the country. And in our ENABLE-2 or phase 2 clinical trial that's now going on at the moment, we're actually going to open additional clinical trial sites in both Auckland and Christchurch, which will help us to recruit more patients and make this more accessible throughout the country. And so this is just a little glimpse of what it looks like inside our good manufacturing practice lab, where our CAR T cells are made for patients in these high quality clean rooms where the operators have to wear a lot of protective equipment, protective clothing, and there's lots of different pieces of equipment used. So this is a very laborious and time-consuming and expensive process, and it also means that we can only make one patient's T cells in the lab at a time. So it's not very efficient for scaling up. So towards the end of our phase 1 trial, we worked together with a startup company, BioAura, who were incubated within the Malaghan Institute and are now developing to be contract manufacturers with a new manufacturing site planned in Christchurch. And so their role will be to produce CAR T cell therapies on a large scale, both for the Malaghan and perhaps for other partners, while the Malaghan Institute will continue to do our basic research here in Wellington and to run new clinical trials of future CAR T cells. And so what we did when we automated our CAR T cell process was to use this machine called the Lonza cocoon. And it's this little white bubble here that has a plastic box on the inside, which is a sealed compartment in which we can grow the CAR T cells completely safe and isolated from the outside environment. So that keeps them very clean and high quality. And the fact that we can put many of these machines into one room and have the same number of operators running them all at the same time means that we can be a much more efficient and quick in treating lots of patients with CAR T cells. So the final results of our ENABLE-1 clinical trial were that there was very good safety and efficacy, or good response rates. So we compared our trial, which is on the right here, with the 3rd generation CD19 CAR T cells, with a trial that was done with the leading 2nd generation CAR19 T cell product in the US. And the main difference here was just the addition of this TLR2 domain. So we examined what number of prior therapies and stem cell transplants patients had had in the 2 trials before getting CAR T cells, and those rates were slightly higher in the New Zealand trial. We also looked at any side effects or severe toxicities, and if you look at all these numbers in the right-hand column, they're all lower than in the US trial. And most importantly, there were no severe inflammatory events, either the systemic ones or the neurotoxic ones. So that was a really great safety profile. And finally, the efficacy or the complete response rates, where there was no more detectable tumor when patients were analyzed at 3 months, was almost identical to this lead therapy overseas. So this is really encouraging data, which led us to be able to start our new ENABLE-2 phase 2 clinical trial, which started in about August of this year, and we aim to recruit 60 patients over the next 2 years. So another research project that we have in the lab to continue to make CAR T cells even more applicable and safer is to use the CD20 safety switch, which allows us to detect CAR T cells better in the blood. So if the patient has CAR T cells and we want to track them in the blood of patients, we've added this extra tag to the surface of the CAR T cells along with the CAR. And we can use a standard hospital test to detect CAR T cells. So a doctor can order a test for their patient and find out information very quickly using this tag. The tag also works with a licensed drug called rituximab, which can delete all the CAR T cells that express the CD20 tag if they became a dangerous risk for the patients. And so this kind of safety tag is not designed for our current clinical trial of lymphoma patients, because some lymphoma patients already receive rituximab for their cancer treatment, but it will be a very useful tool for new CAR T cells against multiple myeloma antigens or solid tumors. So another research project we have in the lab is to address one of the problems of current CAR T cell manufacture, which is that it's quite a long process, taking 10 to 14 days. So that means that patients have to wait a long time for their therapy, but also that the CAR T cells start to get a bit overtired from being grown for so long in the laboratory, and they become what we call exhausted and not very responsive, even before we inject them into the patient to fight the tumor. So we want to address that in two different ways. We want to shorten the manufacture to make so-called faster CARs that are less activated and ready faster, or we want to knock down the exhaustion signals that exist inside the CAR T cells to prevent them from getting this unresponsive behavior when they encounter tumors. And so we will use inhibitory RNAs to target these exhaustion signals. So we have yet another project in the lab. We've got lots of exciting research going on, which is the Kaupapa Māori research project. And this involves one of our PhD students, Danielle Sword, who's going to learn how to make CAR T cells using our current methods, talk to tikanga Māori practitioners and integrate some of their advice to make new and improved CAR T cell production processes, and also get some feedback from patients in their whānau to hopefully integrate all these pieces of information together and finalize a Kaupapa Māori CAR T cell therapy framework, which will be of benefit to Māori and all New Zealanders who are going to receive CAR T cells in the future. So last research project I'm going to talk about today, and I'll hand over to Puja to talk to you more about this, is about dual CAR T cells and how we can use them to target more complex and diverse cancer types. And so tumors or cancers, because they arise from mutations, can continue to mutate as we treat them. And some cancers can contain a lot of different cell types. And some of those might not express the antigen or have the antigenic protein that our single CAR recognizes. So if we have two different CARs, two is better than one, and that will allow the CAR T cells to recognize more different cancer cells directly. And as they kill the majority of the tumor, they may also be able to kill the remaining bystander tumor cells that don't actually have the CAR target antigens. So we think this is going to be another way to improve the effectiveness of CAR T cells in the future. So I'll now hand over to Dr. Puja Paudel, who will tell you more about our research at the Malagin on myeloma.
Thank you, Rachel. Kia ora, everyone. I'm Puja, and I work as a senior research officer in Malagin. So I've been working here for the last 2 years. Before that, I was doing my PhD in Otago, and before that, I was in Nepal. So I came here for my PhD in year 2017. So today I'm gonna talk a little bit about like what I'm doing here at Malagin, and I'm gonna show you some results, like some cool results that we have from our research. So talking a little bit of CAR T cell therapy, as Rachel has explained pretty much everything. So there are overall like total 6 FDA-approved CAR T cell therapies in the market, and 4 of them target CD19 for lymphoma and leukemias, and 2 of them targets BCMA antigens for the multiple myeloma. And these are doing like these 2 BCMA CAR T cells are doing really good, really well in treating multiple myeloma, and with the initial response higher than 90%. But there are still lots of room to improve, and there are still some challenges. And one of the major challenges is the long-term remission. So although they had the most of patients there who are treated with this BCMA CAR T cell therapy, they have a high response rate, but within a year or two, most of the patients relapse. Talking about CAR T cell challenges for CAR T cell therapy, it can be divided into like 3 major groups. So one is being access, because it is personalized therapy. It comes with high cost and also long manufacturing time. And the second one is the adverse effect. It comes with like some severe adverse effect like cytokine release syndromes and neurotoxicity. And also, again, as I said, it comes with like lots of cases, we have seen relapse and resistance in this with these therapy. And here at Mulligan, we are working to address all of these 3 different areas. So we're making faster cars, as Rachel said earlier, that will address the long manufacturing time. And we're making safety switch, which will address the adverse reaction. And finally, the project that I'm working on is dual CAR T-cell therapy, which will address the relapse and resistance in CAR T-cell therapies. So the question is, how is dual CAR T-cell address this issue, like the issue of the relapse and this issue. So to understand that, we need to understand how CAR T-cell and tumor cell interaction works. So here we make CAR T-cells, program them to recognize the tumor antigen in the tumor cells. So we choose the antigen that are really well expressed, and our CAR T-cells will easily recognize those tumor cells when they go into the patient body. But over the time, these CAR T-cells are very tricky, and they have like basic instinct of survival. So they can do whatever they can to for survival. And in this case, what they can do is they can hide out the antigen target that our CAR T-cells are programmed to recognize. And they can reduce, like one thing they can do is they can reduce it down so that our CAR T-cells will have very difficulty to find those tumor cells. And they also can change it completely to something else, which our CAR T-cells are not programmed to recognize. And the next challenge is the tumor heterogeneity. So in this case, the CAR T-cells, the tumor cells might have, like from the beginning, might have different types of antigen on their surface. For example, some of them will have like A type of antigen, some of them will have B and C. So our CAR T-cells are well designed to target the A type of antigen, but they are not designed to recognize B and C. So what happens here is they can easily kill the A type of antigen presenting tumors, but they can't kill the other ones. So the other ones that are not killed, they will come back, and that will cause the relapse in patients. So to answer all this, our dual CAR therapy is one of the best strategy to mitigate this antigen escape and tumor heterogeneity. So here we introduced 2 CARs in the T cells that will like recognize 2 separate antigens at the same time. So the idea is 2 is better than 1. If one of the target is lost, or the tumors are like able to hide that one target or one antigen, then we have the second one that could be with the backup for our CAR T cells. And because we have 2 antigens, we need to figure out like what could be the best combination to make that dual CAR T cell therapy. So these are all the potential antigen that are expressed in multiple myeloma cells that could be used against them to make CAR T cells. So most of them are most of them are being researched, and some of them has already made into the clinical trials, and some of them are still in preclinical stage. So the one that are in this highlighted boxes, they are all already have made it into the clinical trials, and BCMA is the most successful one, like with already 2 approved therapies. So the reason that BCMA is most successful is like they're abundantly expressed in the multiple myeloma cells, and they are not expressed in healthy cells. So these are the 2 criteria that we need to think about, 2 main criteria that we need to think about when we make CAR T cell therapy against any antigen. So apart from them, like the GPR, C5D, and CD19 are the other 2 good targets for CAR T cell therapy, and lots of clinical trials are going on, like with different combination of these antigens, CAR T cells against these antigens. So, and for our study, we decided to go with the combination of CD19 and BCMA because we know these 2 therapies are already approved and they are doing really well in clinical. So we decided to put 2 really well doing CAR T cells together so that they make better dual CAR T cell therapy. So how we make our CAR T cells in lab. So basically, it's pretty much similar to what happens in clinical trials. But instead of using patient blood, we use healthy donor blood. And from that blood, we get the blood cells out. And again, we select the T cells out. And then these T cells are then reprogrammed with the lentivirus that carries the genetic information or DNA information to make the CARs. So once they go into the cells, our cells will process those information to make the CAR T cells. And instead of these cells, instead of going into the patient in clinical CARs, we use them to do different experiments to make sure that these CAR T cells are really good at killing, if they are good or not in killing, and if they are safe or not. So this is how we make our dual CAR T cells. So this is our T cells, and we put them together with the lentivirus that has information for CD19 CARs and the lentivirus that has information for BCMA CARs. We put all of them together so that they make our nice dual CAR population. And after we make dual CAR T cell cells, we check their CAR percentage, like how many CARs are there in our production. So we use this flow cytometry method to determine how many CAR T cells we have. And basically this can show us in this little box, you can see here, this is a single CAR that we get from that population. This is BCMA CARs, this is the CD19 CAR, and then this center population is the CARs that has both the antigen CD19 and BCMA. And once we make our CAR T cells, we need to know, are they any good? Are they good at killing tumor cells or not? And then if they are really good at killing, are they working better than single CARs or not? So if they are not working better than single CARs, there is no point on making dual CAR T cells. So to answer these questions, we do some experiments where we put CAR T cells and tumor cells together. And we monitor these cells, putting them into this little machine called Incuside, and that will take pictures every few hours. And we can check the pictures and see what's going on with the tumor cells. And I can show you some cool videos that we have of these. So here, the red dot you see is the tumor cells, and you can't see the CAR T cells because they are not fluorescent colored, but the tumor cells are fluorescent red, so they can be detected with the fluorescent microscopy. And so the first one is the tumor cells with T cells, and this is where you see the tumor cells are growing without anything to control. So they're in several days, in 5 days time, they will fill out the whole plate. But when we put the tumor cells with CAR T cells, what you can see here is we see less green, less red color, and the green colors are the dye that will indicate the death of the cells. So all the dead cells are colored greens. So you can see we have eliminated pretty much all of these tumor cells from here. So that kind of suggests that our CAR T cells are doing really great in killing tumor cells. So there's another experiment that I did. So basically the same thing that we did earlier, but this has been magnified by 60 times. And you can see really big individual CAR T cells and tumor cells. So here the pink colored cells are the tumor cells and the green are the CAR T cells. And you can see how tumor cells are getting attacked by the CAR T cells, and they're pretty much disintegrated or become something weird shaped from their nice round shape. So this video was taken for an hour, but we could probably see a lot better completely vanished tumor cells if we take this video a little longer. So these all kind of suggest that our CAR T cells is really good and it's working, our dual CAR T cells. So the next thing we wanted to check is since our dual CAR T cells are killing tumors, we wanted to check if they are any better than single CARs or not, and if they are any better in overcoming the CD19 antigen loss. So for that, we made this new tumor cell lines that has less CD19 antigen, so just mimicking the antigen loss in the clinical setting. And the other antigen is still quite high. And we put these tumor cells with CAR T cells. And here you can see the CD19 CAR T cells, they are not able to control the growth of tumors. They are pretty similar to when there is no CAR T cells at all. But if you see this BCMA CAR T cells, they are really like killing pretty much most of the tumor cells, and dual CAR T cells are doing even better. So when we put them in this graph, so you can see here, the blue line is the CD19, the tumor cells with CD19 CARs. So these tumors are like growing, and then they are not, they're pretty much similar to when we don't add any CAR T cells into that. And the other two, the BCMA, and this purple one is the BCMA, and the red one is the dual CARs. So at the beginning, both of them looks like they are doing really well. But then after several days, the BCMA CARs, they start losing the control over the tumor cells. This could be potentially like similar to what we see in the clinical setup where the patient relapse within a couple of years. But our dual CARs is doing really great, and then there's still controlling tumors very well until the end of the experiment. So to summarize all of that, so we have successfully developed dual CAR T cells targeting CD19 and BCMA antigens, and our dual CAR T cells perform better compared to single CAR T cells in tumor killing experiments. And our dual CAR T cells can overcome antigen loss in tumors in the lab. So I would like to hand over back to Rachel to talk about more about the future direction of our this research.
Thank you, Puja. So yes, as Puja was saying, we are not stopping here. We have more research planned in the future, and some of our future goals are being supported by some generous philanthropy, which is helpful to supplement the grant funding that we survive on as a research institution. So one of our goals is to test some new, either single or dual CAR myeloma-specific cells in the lab, and develop the most promising candidates as a CAR T cell therapy. And so we want to try the existing antigens and CARs that Puja has been showing you, and also some new ones that we'll develop together with some of our scientific colleagues. We want to focus on some of the other points I introduced earlier in my talk, which is to improve CAR T cell manufacturing so we can make CAR T cells and deliver them to patients more rapidly. And we also want to silence some of those exhaustion signals that inhibit the ability of CAR T cells to properly combat tumors in the patients. One of our major goals is to develop a phase 1 clinical trial of new CAR T cells developed at the Malaghan Institute to treat myeloma in New Zealand. And our goal is to do this within 4 to 5 years. But possibly even before that, there might be the possibility of other CAR T cell trials coming to New Zealand, thanks to the fact that in our ENABLE-2 phase 2 clinical trial, we're opening more sites for CAR T cell clinical trials in Auckland and Christchurch. And when more hospitals in New Zealand have their expertise to deal with CAR T cells, that might allow some other international CAR T cell trials to come to New Zealand. And finally, we want not to stop at clinical trials. We hope that we're going to be able to bring CAR T cell therapies to New Zealand as a standard of care therapy for cancer, and the Malaghan Institute will continue to support that endeavour. So lots of people to acknowledge who have been involved in this work. Most importantly, the clinical trial participants in the whānau who have generously agreed to be in our clinical trials and to donate some of their cells for research. Everybody at the Malaghan Institute, who are numerous, who've contributed to this project, our partners at BioAura, and colleagues in China, at Te Whatu Ora in both Wellington and Waikato, our trial safety committee, most thanks to our funding partners also, and especially the David Levine Foundation, who has funded a myeloma CAR T cell research plan of up to 5 years to try and get myeloma CAR T cell therapy from New Zealand into the clinic here in New Zealand. Finally, thank you again to Myeloma New Zealand for hosting us. And we are now available to take any questions. If there's anything we're not able to answer for you here today, here's some information how you can reach out to the Maligan to find out more, or if you'd like to do something to be involved in our work as well. Thanks very much.
Thank you very much. So if I start off with some of the questions that we've got. So first question is, for the phase 2 clinical trial, is there an age limit or cut off?
So there will indeed be an age limit. There are exclusion and inclusion criteria for all clinical trials to make sure that the safety of patients is guaranteed. As I'm not a clinical doctor, I'm a laboratory researcher, I can't give you the exact details, but there will be some more information on our Maligan website, or you can reach out to some of the people listed on the slide to find out more.
Thank you. Next question. When will CAR T therapy be available for paying patients in New Zealand?
So that's not something I'm sure about either, but possibly Bio-Aura, with their endeavours to set up their Christchurch work and facility, they would be able to answer that in the short term future.
Thank you. Next question. Does CAR T cell therapy result in significant immune suppression in the patient after treatment?
That's a very good question. CAR T cell therapies are associated with what we call cytopenias, or lower numbers of blood cells, including immune cells. And those cytopenias can be short-lived, which is normal because there's been a bit of chemotherapy and then CAR T cell activation, or they can be longer lasting, and the longer lasting ones can actually be quite severe problems. In our New Zealand Enable 1 CAR T cell trial with the 3rd generation TLR2 CARs, we actually saw a lower proportion of those longer term cytopenias, so maybe another indication of the safety of our CAR T cell therapy.
Fantastic. Next question was, what a fantastic presentation. Thank you for your amazing work. Can patients go through CAR T more than once as new generations develop?
That is a good question as well. If you have had a CAR T cell that uses sort of the exact same CAR structure, particularly the antigen recognition part on the outside of the cell, giving another CAR T cell with that same antigen recognition domain could be problematic because your immune system might recognize it and reject it. But you could have one kind of CAR T cell therapy and then have a different kind of CAR T cell therapy, or some patients have a BiTE therapy first and then a CAR T cell therapy later, or the other way around.
Great. And just another person saying it was a fantastic presentation. Next question, fantastic work. Which myeloma cell line was used, and what fluorescent dyes or probes were used to indicate the tumour in the purple and a CAR T in green?
All right.
So for this one, we use K562 cell lines, and for the fluorescent dye, we use mCherry. Like there's a fluorescent protein, so we put that we transduce the cells with the fluorescent protein, so they're like all the time they're red.
So yeah, we can find some fluorescent dyes in nature from things like jellyfish or fireflies or other sort of natural sources of fluorescence, and we can engineer those genes into our tumor cells to be able to detect them. And the K562 tumor cell line that we use in these experiments is a type of blood cancer, but it's not specifically a myeloma. We also engineer the myeloma antigens into that cell, so it's a laboratory model only.
Great, thanks. So I've got another question, but just a reminder, if anyone does have any questions, you can put them in the chat. There's the little, the Q&A bit along the bottom. So next question is, why, when these trials have been conducted overseas, do we need to do trials? Why can't we use the already existing overseas treatment?
That's another question that I think you probably need a clinical doctor to give you the real answer, but I believe it's because in New Zealand we want to be very careful that what we give to our patients has been evaluated and tested by us to be safe for the New Zealand population, and also because we have some quite strict and careful regulatory requirements for clinical therapies to protect the safety of our patients. So we want MedSafe, our New Zealand regulator, to be sure that the clinical trial is of good quality and safety before we launch it here.
And would it also be correct to say, when the question is why, when these trials have been conducted overseas, what you're doing hasn't been conducted overseas because what you're doing is slightly different? Is that? That's
a good point. The CAR T cells, well, the new CAR T cells we're developing at the Maligan have not been tested anywhere else before, but I think MedSafe would also want to evaluate international products here in our local population as well.
Right. Next question. Does solid tumour dissolved in dual CAR? Oh, does solid tumour involved in dual CAR-T therapies, how are we able to distinguish the trials? How can we approach to get in any intern positions to learn about Maligan?
Yeah, so the Maligan doesn't actually have any solid tumour trials yet, but we hope to move into that space in the future. The best way to find out more is to contact the Maligan, and then we've got some experts that can explain the differences. Solid tumours are a little bit more difficult to treat with our current CAR-T cells, and that's why both the methods that I was talking about and the methods Puja's talked about will enhance the ability of CAR-T cells to fight those more difficult tumours, and we hope there'll be lots of progress in the next few years.
Great, thanks. And then a question about intern positions. I think from memory, Maligan has a recruitment page on the website, and people would be able to look there for any vacancies that you've got.
Yep, that would be a great place to look. And any university students who are interested, we take summer interns during the sort of university holidays who can get an experience of the lab as well.
Awesome. Great, thanks. And do those intern positions go up on the recruitment page?
I think they do, but universities generally advertise the possibility of getting funding for internships, and then students are welcome to reach out to any organisation that does research and see if they have some space. That's
great, thank you. Next question. Are there issues for high-risk multiple myeloma patients with gene mutations, copies of IQ gain, etc., and CAR-T cell therapy? So that's, again, maybe a little bit out of my field of expertise, but CAR-T cells, because they recognize a specific protein on the cell surface, as long as the mutation that a myeloma patient who's high risk has isn't in that particular protein, there shouldn't be an issue. That's great. Were there any other questions? That looks like the end of the questions at the moment. Did we have any other questions? I'll just have a little bit of time for people to put in the Q&A. That might be the end of the questions. Was there anything else that either of you two had that you wanted to cover off, or? I don't think so. I think we've probably talked about everything. All right, well, that's great. Well, we'll end it there unless someone pops. Oh, here's another question. So one question is, it's a couple more actually. It's 330K about in China. So this is kind of a comment. So we really hope that you progress your work and have it available here soon, and thank you for your work. And another person said, thank you very much to you both. And it's an amazing presentation. And I'm sure everyone watching has really similar thoughts. Thank you very much. That's much appreciated feedback to hear. And it's very nice to have members of our community be interested in our research. That helps motivate us to do the work that we do. Right. And just there's a comment here from Maligan, just to clarify, the reason that Maligan are trialling it here is to help set up the regulatory and clinical landscape to develop this in New Zealand. Right. And someone said 4 to 5 years seems a long time away. And I guess that's because as you've gone through, there's so much to it that you're doing. Yeah. That's right. So the experiments that Puja has shown are very exciting, but they're still some of the first experiments that we've done in the lab. And so there's lots more of safety and quality testing that we need to do before we're ready to put something into patients. So yes, maybe 4 to 5 years for an internal Maligan trial. But as I was saying, and as the Maligan were confirming, the fact that with our first enable trials for lymphoma, we've been setting up all the clinical machinery and regulatory background that's needed for other CAR-T cell trials. We hope that'll bring more of them from other places as well, not just the Maligan. All right, well, I think that might be, unless we have another flurry of questions, that looks like it might be the end of the questions. I'd just like to thank you both so much for putting together this presentation and talking to us today. I found it personally just absolutely fascinating, and especially knowing that this work is, I'm in Wellington, knowing this work is going on in Wellington. So yeah, thank you very, very much. And yeah, we'll leave it there. So thank you. Right. Thank you, and we hope to catch up again soon. Great. Have a good day, everyone. See you later. Bye. Bye.